to all. Today we are going to discuss about the virus. And this virus is a very important topic. And very first you must know that virus is a Latin word. And the literal meaning of this word virus is poisonous fluid, or we can say it as venom. Okay. There was a scientist whose name was Ivanovsky, who separated a microorganism from the sap of the infected plant and named it as tobacco mosaic virus, that is known as the TMV. He reported that viruses are smaller, in fact they are smaller than the bacteria also, and they can pass through the bacteria proof filters. The real credit for the discovery of the viruses goes to the scientist known as the Ivanovsky. There was one more scientist whose name was Bacteriophages, who called them as contagium vivum fluidum. And what is the literal meaning of this term contagium vivum fluidum? Means living infectious fluid. Means Bacteriophages called them as living infectious fluid or contagium vivum fluidum. Okay, and Stanley was the scientist who crystallized the TMV virus. Stanley was the scientist who crystallized the TMV virus first time, and hence for this discovery he was actually awarded the Nobel Prize also. There were two group of scientists, that is known as the Borden and the Perry. Borden and the Perry studied the chemical nature of the viruses and said that these are nucleoproteins, means they are nucleoproteins. This was said by two scientists, Borden and Perry. Okay. Now when we are talking about the viruses, we must know about some characteristic features of them. Means they are submicroscopic. Very first point is that they are submicroscopic and they are non-cellular organisms, and they are smaller than 200 micrometers. Again I am repeating, they are submicroscopic and non-cellular organisms, and they are smaller than 200 micrometers. They are obligate parasites. What do you mean by obligate parasite? Obligate parasite means they can live inside the living host only. They are not having any other option for life other than parasitism. When they are inside the body, they are living. Outside the body, they are acting as non-living creatures. They have either DNA or RNA. This is very important point. No virus is having DNA and RNA both. They are having either DNA or RNA. There are few viruses which are having DNA as the genetic material. There are others who are having the RNA as the genetic material. They are so much small that they can pass through the bacteria proof filters also. They are smaller than bacteria. Okay, and they are inert. They are inert outside their specific host cell in crystalline form. Means outside the body they are not at all active. They are acting as inert structures. Okay. Now we must know that viruses form a transitional group between living and non-living creatures. What I am saying, viruses form a transitional group between living and non-living organisms. Means we can say it as that they are having some non-living characters and some living characters. At least when we are discussing about the viruses, we must know that what are the non-living characters of the viruses and what are the living characters of the viruses. See here, non-living characters, they are having no protoplasm. Like a cell, they are in the cell, protoplasm is there, but they are having no protoplasm. They are having no enzyme system. They are having no power of respiration. They can be crystallized and they do not grow in culture medium. Okay, again I am repeating, the non-living characteristics of the viruses are no protoplasm, no enzyme system, no respiration. They can be crystallized and they do not grow in the culture medium. These are the non-living characters. But what are the living characters? See here, because we know very well inside the body they are acting as living creatures. So they contain nucleic acid, as a result of which they are capable of synthesizing the proteins. As they have nucleic acid, we know very well that they are nucleoproteins. They are having nucleic acids, and as a result of which they are capable of synthesizing the proteins. It is a living character. They can multiply inside the host cell. They cannot multiply outside, but they can multiply inside the host cell. When they are inside the host cell, they are having the power of multiplication. And to prove this thing that they are living characteristic, the point is that they causes diseases. As we know very well, there are many viral diseases which are actually caused by the viruses, means they cause diseases. Okay. Now, when we are talking about the viruses, we must know about the classification of the viruses. There was a scientist whose name was F.O. Holmes. F.O. Holmes was a scientist who actually divided the order Virials into three suborders. Okay, the very first suborder was Phytophagini. Phytophagini. Now, what is Phytophagini? Phyto means what? Plants. Virus infecting plants. Those viruses which infect plants are included in the Phytophagini suborder. Now, Zoophagini, we know very well, Zoo word is actually coming from the animals. So, viruses which infect animals are included in a suborder Zoophagini. And Phagini, the word Phagini is used for Bacteriophages, means viruses, that is the Bacteriophages which infected the bacteria. Phagini means the viruses which are infecting the bacteria. We know very well that viruses which infect bacteria are called as the Bacteriophages. So viruses infecting bacteria are included in a suborder known as the Phagini. Okay. Now let's talk about the structure of the viruses as well as the morphology of the viruses. Now let's have a look on the morphology and the structure of the viruses. First of all, we'll be talking about the size. Now, viruses are very small, we know, and they are less than 200 micrometers, okay? But the largest virus is treated as a smallpox virus, which is also called as the variola virus, and it is 250 micrometers. While if I talk about the smallest virus, so the smallest virus is the 10 micrometers in size, and it is the foot and the mouth disease virus. This is the size of the virus. Now, if I talk about the shape, now shapes are very much variable, okay? Shape may be spherical, may be rod shape, may be tadpole-like. Now, generally the spherical shape of the viruses are the influenza viruses, and rod shaped viruses are the TMV, tobacco mosaic viruses, and the tadpole-like shape is of the bacteriophages. Okay, so we have talked about the size, we have talked about the shape. Now, if we talk about the chemical composition, so if I talk about the chemical composition, viruses are made up of two things. One is known as the nucleic acid and another is known as the protein coat. If we see any virus, we will be able to see two important ingredients. One is the nucleic acid and another is the protein coat. Just now we have discussed that no viruses are having both the DNA and the RNA. Either they will be having the DNA or either they will be having the RNA. Now, generally what we see, generally I am not talking perfectly, generally in plant viruses, single-stranded RNA present. SS means single-stranded RNA is present, but there are few exceptions. In the cauliflower mosaic virus, now cauliflower mosaic virus is also plant virus, but it is not having the single-stranded RNA. Instead, it is having double-stranded DNA. It is having double-stranded DNA. DS means double-stranded DNA. Now see the reverse thing. We have said that generally in plant viruses, single-stranded RNA present. But generally in animal viruses, double-stranded DNA is present. But there are some animal viruses where RNA is present. I have quoted at least 4 or 5 examples. Influenza virus is having single-stranded RNA, though these are all the animal viruses, but still they are not having the DNA, they are having RNA. Influenza virus is having single-stranded RNA. Raux sarcoma virus is having single-stranded RNA. AIDS virus is having single-stranded RNA. Okay, poliomyelitis virus is having single-stranded RNA. But I have quoted 2 examples here. Wound tumor virus, okay, and reovirus. Both wound tumor virus and reovirus, they have double-stranded RNA. Okay, so you must know about the animal viruses as well as the plant viruses, and you must know that what type of the nucleic acids are present in them. Now, if we talk about the AIDS virus are called as the retrovirus. And they copy RNA into DNA. Normally what happens? DNA forms RNA, but they copy RNA into DNA, and means they are showing reverse transcription, and they are showing reverse transcription with the help of the reverse transcriptase enzyme. Okay. Now in bacteriophages are also type of viruses which affect the bacteria. So in bacteriophages generally DNA is present. In bacteriophages generally DNA is present, but here there are some exceptions. Say for MS2 bacteriophage, F2 bacteriophage and R17 bacteriophage, right? They are having the genetic material as RNA. They are having RNA. Instead of DNA, they are having RNA. Now, if I talk about the total number of the genes in the viruses, so the number of the genes are not fixed in the viruses. If I talk about the large virus, say for vaccinia is a cowpox virus, so in it approximately 100 genes are present. But if we talk about the small viruses, right, then they are only having 3 to 4 genes. Now, let's talk about the second component, and that is known as the protein coat. Now, the protein coat of the viruses is called as the capsid, and it is made up of many small units. And these small units are called as capsomeres. Okay. Now, the central core of the virus along with the capsid, is called as what? Nucleoplasmid. That is capsid. Why? Because nucleo means what? Nucleic acid, and nucleic acid may be DNA or RNA. So nucleocapsid. And not only this, an additional covering is also present in some viruses around the capsid. Means outside the capsid they have an additional covering, and it is called as a limiting membrane, or it is called as the mantle, or it is called as the envelope. And such viruses are also called as enveloped viruses. They are also called enveloped viruses. Okay. Now, if we talk about the symmetry, so there are different types of the symmetry found in the viruses. Say for the helical symmetry, means the capsomeres found in the capsid are arranged in helical manner. Examples are tobacco mosaic virus, influenza virus, all have helical symmetry. Now, cubical symmetry means capsomeres are arranged on the surface to form a 20-sided cube, and hence they are having the cubical symmetry, such as for tobacco, that is known as the turnip mosaic virus. And bisymmetrical or mixed symmetry is found in the bacteriophages. Okay, so this was all about the structure and morphology of the viruses. Now we'll be talking some more details of the viruses, and that are the bacteriophages. Especially we will be emphasizing on the bacteriophages. Now we will be talking about a very important virus, and that is known as the bacteriophage. As we know very well, bacteriophages are the viruses which infect the bacteria, and they were first of all discovered by two scientists named as Stuart and Harrell. Okay, and bacteriophages are generally having the genetic material as a double-stranded DNA. Means in bacteriophages mostly double-stranded DNA is present, but there is exception. I have also talked earlier that there are few bacteriophages where the DNA may be single-stranded, and Pi-X174 bacteriophage has single-stranded DNA. Okay. Now, if I talk about the structure of the bacteriophage, a bacteriophage consists of two important parts. One is known as the head, and another is known as the tail. And in between the head and the tail, neck and collar are present, and tail is surrounded by tail sheet. Okay, and head consists of two parts. One is known as the DNA, and DNA is surrounded by a protein coat. As we know that generally DNA is present in the bacteriophage. So head is prism-like, it is hexagonal, nucleic acid is DNA surrounded by the protein coat, and the size is 950 angstrom into 650 angstrom. Means these are the length and the breadth. Okay, and the tail is having the size of 950 angstrom. It is its length, and tail consists of the tail fibers, and each tail fiber is 15 angstrom in length, and total there are how many tail fibers? 6 tail fibers. Okay, so this is the structure of the bacteriophage, which seems to be just like a tadpole, and it is prism-like head. It is having prism-like head. Okay. Now, based on that, whether the bacteriophage is actually affecting the bacteria or not, we can say that the bacteriophages are of two types. One is known as the non-virulent and another is known as the virulent. Okay, non-virulent, as the name is indicating, does not affect the bacteria, and virulent is infecting the bacteria, and that's why it is called as non-infective phase. It can also be called it as prophase, and it is also called as the lysogenic phase. And the another one is known as the virulent phase, or we can say it as the infective phase or the lytic phase. Okay, the non-virulent phases are the phases which don't cause lysis of the bacteria, means they are not responsible for the breakdown of the bacteria, and the virulent phases are the phases which cause lysis of the bacterial cell. Okay, and if we talk about the applications of the bacteriophage, so we must know that in our India we are having a holy river Ganga, and the purity of the holy Ganga is maintained just because of the bacteriophage, which are the viruses which are infecting the bacteria. It is also used in the control of bacterial diseases because it is having the lysis property, and that's why the bacteriophages are also used in the control of many bacterial diseases. Okay, it is also used in studying viral infection mechanism. If you want to study the viral infection mechanism, that how do the virus infect, okay, then we can you take use of the bacteriophages. Okay, so this was all about the bacteriophage virus, and we must know about a virus known as the tobacco mosaic virus. It is very important, okay, and many times the question is asked, that is tobacco mosaic virus. Tobacco mosaic virus was discovered by Ivanovsky in the year 1892. As we know that it is having the helical symmetry, it is rod shape in structure, it has the genetic material single-stranded RNA which is having 7,300 nucleotides. Its size is 300 nm to 20 nm. This size 20 nm may be 18 nm also. Okay, in some books you can get the size of the TMV virus 300 nm into 18 nm also, so don't be confused. It is varying between 18 to 20. Okay, now number of capsomeres in capsid. We know very well that the protein coat is called as the capsid, and the subunits of the capsid are called as the capsomeres. So the number of the capsomeres is 2130. Okay, 2130. Remember this number because question is asked, 2130. And if we talk about the TMV, then it consists of 5% RNA and 95% of the protein. Means in the entire TMV, the percentage of the protein is 95% and the percentage of the RNA is 5%. Okay, this is the structure you can see. This is the central lumen. This is the RNA helix, and RNA here is single-stranded. This is the protein coat, and the protein coat is made up of many subunits, protein subunits known as the capsomeres. The number of the capsomeres is 2130. So this was all about the bacteriophages and the TMV. Now we will be talking about viruses, virions, prions, and the diseases caused by the viruses in plants and animals. Now we will be talking about the viroids. Very important one, because many people are having the confusion that what is viroids, what is prion, what is virion, what are interferons. Okay, so let's have a look. Viroids. Viroids were first of all discovered in 1971 by a scientist known as T.O. Diner, and they are smaller than the viruses also. We know very well that smaller than the bacteria are the viruses, and smaller than the viruses are the viroids, and they are new infectious agents. Okay, and they are subviral infectious agents. These subviral infectious agents are called as the viroids, and the special feature of the viroids is that they contain single-stranded RNA, but they are not having the protein coat. Normally what we see, normally the viruses are having the genetic material DNA or the RNA along with the protein coat, but the viroids are having, what you have to remember is that viroids are having the single-stranded RNA as a genetic material, but they are not having the protein coat, and they are causing a disease known as the PSTV. PSTV means potato tuber spindle disease. Potato tuber spindle disease. Potato tuber spindle disease. Okay, spindle disease. Okay, so the next one is the prions. Prions are known as the slow viruses. They were discovered by Stanley B. Prusner, okay, which contain only the proteins. Now see the big difference. In the viroids, genetic material was present while the protein coat is absent. here only proteins are there, means these are the viruses which are not having the genetic material and they cause diseases in the animals. Say for they can cause a Kuru disease of the humans. Okay, this Kuru disease is the disease related to the nervous system, okay, in the case of the human beings, and the scrapie disease of the sheep and the goats, it is also caused by the prions. The third one is known as the virion. What is virion? Virion is the completely assembled virus particle outside the host is called as the virion. Again I am repeating, virion is the completely assembled virus particle outside the host is called as the virion. Now what are interferons? So interferons, there were two scientists known as the Findlay and the McCallum. They reported a phenomenon of the viral interference. They reported the phenomenon of viral interference, in which what happens, the cell which is infected by one type of the virus become resistant to the superinfection by the other viruses. What is viral interference? When a cell is infected by one type of virus, then it becomes resistant automatically by the superinfection of some other viruses. Okay, and this phenomenon was actually shown by the Findlay and the McCallum, and Isaac and Lindman gave the term interferon to the chemical substances which are actually responsible for the viral interference. Okay, and the interferons are actually produced by the cells in mammals. In some cases it is also released in the birds also. So interferons are produced by the cells in the mammals and provide resistance against the viruses. Okay, means when one cell is infected by virus, then it becomes resistant by the infection of the other viruses. Means we can also say that the interferons are antiviral proteins. What are interferons? Interferons are antiviral proteins. Antiviral, they are antiviral proteins. Okay, and that's why we say that the nature of the interferon is they are protein in nature. Okay, now the another one is known as the macrophages. What are macrophages? The myco word is actually used for the fungi. So those viruses which infect the fungi are called as macrophages. They were discovered by a scientist known as the Sinden, okay, in a fungi known as the Agaricus bisporus. They have double-stranded DNA. And the shape of the macrophages is spherical or they are polygonal in shape, right? Now the next one is the cyanophages. Now we know very well cyano word is actually coming from blue-green algae, because blue-green algae are also called as the cyanobacteria. So viruses which are infecting the blue-green algae are called as the cyanophages, and they are having the genetic material as double-stranded DNA. Now the another one is known as the coliphage. Now what is coliphage? Coliphage was discovered by the scientist known as the Harrell, and these are the viruses which are infecting a bacteria, and that bacteria is called as the E. coli. Okay, so you must know about the viroids, prions, virions and interferons. All are important from the examination point of view, and many people are having the confusion regarding this, right? So now at the last we must know that the plant diseases and the animal diseases. There are many plant diseases and many animal diseases which are actually caused by the viruses. We will be not talking in details in this video about the diseases. I have just talked about some names of the diseases which are actually caused in plants as well as animals. For viewing the details of these diseases, you can watch my next videos. Now see, the plant diseases are tobacco mosaic disease, leaf curl of papaya, yellow vein mosaic disease of lady's finger, tomato leaf curl, tungro disease of the rice, bunchy top of banana, and potato leaf roll diseases are some of the diseases which are actually caused by the viruses in the plants. And there are many animal diseases also, especially in the case of the humans. There are many diseases which are actually affected by the viruses. Say for AIDS, the most common dreadful disease which is also called as the death warrant, right? And it is a growing disease, and many people in the world are actually suffering from this disease. And AIDS is caused by the retrovirus, we know very well, right? You can see the detailed videos. Chicken pox, which is caused by the varicella virus, smallpox is caused by the variola virus, and measles is caused by rubella virus. So student, this video was the complete video based on the viruses. Keep watching my videos. Please like and share my videos and subscribe my channel. Thanks a lot.